Hello, students. Have you ever wondered how your body knows how to make proteins? Think of your body as a factory constantly assembling proteins, which are like the building blocks of life. Just like a recipe needs ingredients and instructions, your body needs a guide to assemble proteins. And that guide comes in the form of mRNA. Translation is the fascinating process where the instructions in mRNA are read by a ribosome to create proteins. It's like a tiny assembly line working non-stop in each cell. Let's take a closer look at how this happens. The first step of translation begins in the ribosome, our molecular factory. But how does the ribosome know what to assemble? That's where tRNA comes in, which we'll explore next.
Now, let's dive into tRNA, or transfer RNA. Picture this. tRNA is like a delivery truck, carrying essential ingredients to the ribosome for protein assembly. Each tRNA molecule is specific to one amino acid, the building blocks of proteins. The tRNA reads the mRNA code, like a worker reading instructions on a blueprint, by matching its anticodon with the mRNA's codons. Just like how you can only fit the right key in the right lock, tRNA only pairs with its matching codon in the mRNA. As you can see in this diagram, tRNA has an acceptor arm where it attaches the amino acid it carries. This precision is crucial for making sure the proteins are assembled correctly. So, each tRNA delivers a specific amino acid to build our protein chain, one piece at a time. Now, let's visit the real factory, the ribosome. This molecular machine is composed of two subunits, like a two-part puzzle that comes together to start the translation process. The ribosome reads the mRNA sequence and, with the help of tRNA, it assembles the protein. It moves along the mRNA, linking amino acids in the correct order, much like how a factory conveyor belt moves parts to be assembled into a finished product. And here's something cool. The ribosome isn't just a passive reader. It actively helps to form peptide bonds between the amino acids. This bond formation is the key to building a protein. Imagine it as a factory worker using a special tool to weld pieces together, creating a strong, functional chain. This is how your cells create proteins, which carry out nearly every function in your body. Now, let's break down translation into its key stages: initiation, elongation, and termination. First, initiation. The ribosome binds to the mRNA at the start codon, AUG, and the first tRNA brings in the amino acid methionine. It's like the beginning of a new project where the first piece is carefully placed. Next, elongation. This is the main work phase. The ribosome moves along the mRNA and neutrinos bring in their amino acids. The ribosome links them together with peptide bonds, building a long chain of amino acids. It's like the assembly line picking up speed, adding pieces one after another. Finally, we have termination. When the ribosome reaches a stop codon, a special release factor comes in and signals the end of translation. The ribosome releases the completed protein chain, just like the final product rolling off the assembly line. At the heart of translation is peptide bond formation, the glue that holds the protein together. Each time two amino acids are brought together, they form a peptide bond. This requires energy, which is supplied by ATP. It's like a worker using a tool to fasten the pieces together. This bond is crucial because it creates a long chain of amino acids, which will eventually fold into a functional protein. Without peptide bonds, there would be no protein structure and no function. Imagine trying to build a Lego structure without snapping the blocks together. It simply wouldn't hold. To wrap it up, translation is the incredible process where mRNA guides the creation of proteins. We saw how the ribosome reads the mRNA code, how tRNA delivers the right amino acids, and how those amino acids are joined together through peptide bonds to form a polypeptide chain. Isn't it amazing how every protein in your body is made in this precise way? This is how life forms the complex molecules needed to carry out essential tasks, from building cells to transmitting signals to fighting diseases. So, the next time you think about how your body functions, remember, it's all thanks to the intricate and wonderfully precise process of translation. I hope you enjoyed this journey through the world of molecular biology. See you next time for more amazing discoveries about the science of life.